As the pathogen, I have gained access into the body. Now I need to find other cells to infect so I can survive and reproduce. I'll just be here in the bodily fluids of this body, ready to infect cells.
This looks weird. As the macrophage who loves eating dangerous microbes, I should probably eat those just to make sure. You're weird and different. We don't like that in the human body, so I'm gonna have to eat you.
Just because you kill me doesn't mean there aren't more of us pathogens.
This pathogen. Tastes dangerous. Let me try again. Yeah, dangerous. I should warn the helper T cell that this intruder is in our body, ready to invade all of our cells. I'll need the help of my friend, the class 2 MHC molecule.
As the class 2 MHC molecule, I have to bind the antigen of this pathogen to the surface of the macrophage, so that the macrophage can present this antigen to the helper T cells.
Helper T cell, here's an antigen. I need you to spread the word that we need to attack the pathogens that have this antigen.
As one of the T helper cells, let me bind to the T cell receptor of the antigen and listen to whatever it needs to tell me. Oh, I see, you're right. This is one ugly antigen. Let me use your cytokines as well as mine so that I can be activated. Thanks. I'm now activated and should proliferate and multiply to tell B cells about this antigen. Hey B cell. You have a problem and I need your help. An ugly pathogen has entered the body and we need to get rid of it. To do that though, I need to activate you first.
Thanks to the helper T cell, I am now an activated B cell. Now I must differentiate into plasma cells, which are the cells that make antibodies.
Plasma cells.
As one of the plasma cells, I need to make the antibodies that match in shape with these antigens so that they can attach to them and attack them.
Now, as an antibody, I'm looking out for these antigens on the pathogen we need to destroy. OMG, there's a pathogen with the antigen that I'm looking for. I'm going to bind to it, and then my helpers, the complement proteins, can help me destroy the pathogen. So how does an antibody help destroy a pathogen? There are a couple things that can happen. When antibodies bind to the antigen on the surface of the virus, they block its ability to bind to the host cell and infect the cell. Also, when antibodies bind to the antigen, they mark the pathogen for phagocytosis by a macrophage. The macrophage can digest the pathogen and secrete lysosomes to destroy it.
There's also a third way that antibodies can help destroy a pathogen. When they bind to the antigen on the pathogen's surface, they activate a system of complement proteins. These proteins form the membrane attack complex, and they make pores in the pathogen's membrane. This allows water and ions to rush in. The pathogen cell swells and eventually lyses.
OMG, we just destroyed all them pathogens. Hopefully they won't come back anytime soon.
Just in case they do, I have my memory cells that for this specific antigen for the pathogen. I will floating around in the blood in different humors in the blood with those specific antibodies in my membrane. So if pathogens that antigen come back, I will be ready to destroy them with my antibodies.
Woo!
Okay.
That's it.
All right, questions from the audience? Okay, I have a question. What's the connection between a plasma B cell and a memory B cell?
once T helper cells activate B cells, they proliferate and they become plasma cells, and they also, some of them turn into memory B cells for later in case that pathogen, like the antigen, comes back into our body.
And also the plasma cells are responsible for making the antibodies that bind to the antigens.
Okay. And then why is it beneficial to have memory B cells?
Because there's a secondary, the time when the pathogen come again, they have a memory of the antigen so that they can release antibodies as quickly as possible.
So basically, like when the secondary immune response happens, like if that pathogen comes back, once they recognize the antigen, you don't have to go through the whole process of the T helper cell, like finding the shape of the antigen and then telling the B cell what kind of antibodies to make, because there's the memory B cell that already has that specific antibody, so that's going to get rid of the pathogen a lot faster.
So the memory B cell already has the antibody embedded in its membrane that's specific to that particular pathogen. And the thing that kids always kind of get wrong here is there like one memory B cell for every attack?
No.
Can somebody explain why it's faster when there's memory cells? Other than what Sonica just said, which was right. That's a big reason why it was faster.
There's more than one memory B cell. Like they're floating all through the blood. So wherever the pathogen enters, there's like nearby memory B cells.
So there's a higher concentration of these floating around so that the next time there's an invader, there's a much higher chance that that's going to encounter it quickly and boom, set off the pathway of making these plasma cells proliferate and make lots of antibodies. Much quicker response. Initially, when it hasn't seen it, you just have to wait for a chance encounter with a differentiated B cell that just happens to fit. And it might take a really long time for that to happen. Okay, audience, your turn. Lindsay hasn't asked a question. Oh my gosh, that's really close.
What's the difference? So the interleukin-2 activates the IL-2 activates the B cell, but for the cell-mediated, it activates the cytotoxic T cells. So when does antibody-mediated immunity take place, and when does cell-mediated immunity take place?
Well, they're two different processes, and they both happen. Like they're not like one happens in the other one, because that's the body's like two ways of making sure it gets rid of all the pathogens. So first, like it tries and attacks all the pathogens that are like floating around in the blood before they infect the cells, and then for the cell-mediated, they find the cells that are already infected and kill them so that the pathogen doesn't have a chance to spread.
And I also think that for the cell-mediated ones, they're almost specific for the viruses that's going to take over the cell to produce a cell. Like other bacteria, they are independent, so they're going to grow by self. So they have to be their production should be limited using the antibodies, which is going to direct response to those pathogens.
Let's just clarify for cell-mediated. Cell-mediated doesn't just have to start with a virus. That was the example that was used in the last presentation. It can start with a macrophage ingesting as well, an antigen presenting, and helper T, and then they can recruit cytotoxic Ts as well. That can happen as well. So the common denominator, the thing that hooks together both pathways, is the general. Hopefully you'll notice that the helper T is involved in both pathways, and it coordinates both pathways in that way, both in the antibody-mediated or humoral and the cell-mediated. Who else has a question? Wiley?
Can you guys explain again how the antibodies dispose of antigens?
Well, basically, like, they can do a lot of things where if, like, so antibodies are like the Y shape, so they have two ends. If a bunch of the, like, antigens get binded onto the different ends of the antibodies and they come together, they can, like, lysis or something, and they can destroy all of them together. Or they can, like, signal for a macrophage to come, and then a macrophage, like, if you gather, like, a bunch of, pathogens with those antigens together, then it'd be easier for like an active H to get all of them. And yeah.
So the antibodies, like sometimes they act as like sort of like a signal to call the macrophage to eat, and then other times like Makoto is a complement protein, so sometimes they'll just attack the pathogen cell and like create pores and cell lysis. There's a couple different ways.
They can clump pathogens together too, and make it easier for the white blood cells to attack and phagocytosize. I always have a hard time with that word. Phagocytosize. Eat. So that's another way. Is that it? All right, thank you.